and let us see what is there in today's learning objectives. Now, today's learning objectives, we are going to discuss about the cell organelles which are found in the cytoplasm, the plastids, the centrosome and the centrioles, the lysosomes and the vacuoles. So, this cell organelles which we are continuing from the previous episode, we are going to discuss about this main four things: plastids, centrosomes, centrioles, lysosomes and vacuoles. So, let's start with plastids. So what are plastids? Plastids are mainly, plastids are also called the kitchen of the plant. What are they called? They are also called the kitchen of the plant cell. So kitchen of the plant cell, as this term is telling you that it is only found in the plant cell. This is not at all found in animal cell. So kitchen means either it is producing food or it is storing food. So it is only found in the plant cell. So there are three main types of plastids, that are chromoplasts, chloroplasts and the leucoplasts. So the first one is the chromoplast, the second one is the chloroplast and the third one is the leucoplast. So the chromoplast, what is it? It is actually, we can say that they contain fat soluble yellow or orange or red colored pigments. So different colors. So what are these different colors? These are yellow, red or orange colored pigments. These are yellow, red or orange colored pigments. They are actually fat soluble. Okay, and they are mostly present in the colored parts of the plant, that is petals and fruits, and they provide colors to the petals and fruits. Is it clear? So yellow, red, orange, whatever fat soluble chromoplasts are there, they provide color to the flowers and fruits, and they contain the coloring pigments which are also called xanthophyll and carotene. Okay, so they also contain the color pigments called xanthophyll, and we can say that they contain the color pigments xanthophyll and carotene. So xanthophyll are yellow colored pigments. These are yellow colored pigments, and carotene, these are orange red colored pigments. Okay, so orange red colored pigments. Now, when we move on to the next thing, that is the chloroplast. So as the term chloro describes, we can say that these are mainly the green colored plastids. So these are green plastids. They possess the photosynthetic pigments. Is it clear? They contain the photosynthetic pigments and they are green plastids. So green parts of the plants like leaves, the young stems, which are having green colors, these are actually due to the presence of this pigment of plastid called the chloroplast. So other pigments are also contained by chloroplasts. So chloroplast contains DNA, RNA and the various enzymes for the photosynthesis. Is it clear? So chloroplast, these are masked actually into large quantity of chlorophyll only, but they also contain the DNA, RNA and various enzymes essential for photosynthesis. Now the third most important plastid is the leucoplast, which is, I couldn't accommodate in this one slide. I'll move on to the next one. There we will see what is leucoplast. Now these are actually, till now, chromoplasts and chloroplasts, we have seen that they are colorful plastids with orange, yellow, red and other pigments. But these are all colorless plastids. They don't have color. They don't have any pigments. They are actually named on the basis of the substances stored. So mainly what do they store? They store starch. So as we all know that starch is having no color. So it is actually a colorless plastid. So cells of potato have a ton of large amount of leucoplasts. So if they tell you to give an example of leucoplast, so you can say that cells of potato have a ton of large amount of leucoplasts. So if we start discussing about the functions of leucoplast, what we will do? So the leucoplast, we have known that it is mainly helping in the storage of food, main function. Functions of functions of chromoplastis. They are actually the colored plastids which provide color to the flowers and fruits. Okay, so what are the chloroplasts doing? Chloroplasts, they help in photosynthesis and thus help in the synthesis of food. And overall, if we are telling about the functions of plastids, what is it? It is the kitchen room of the cell, or rather the plant cell. I have told you in the first introduction time only that it is actually present only in the plant cell and it is called the kitchen room of the plant cell. Now let us move on to the next cell organelle. It is also a very important organelle of the cell. Let us move on to the next cell organelle. Okay, sorry, before that I would like to discuss about the diagrams. So let us discuss about the diagrams of the plastids here. So what is there in the diagrams? First, we are seeing the diagram of this is actually the submicroscopic diagram of a chloroplast. This is a chloroplast. This is a leucoplast. And this is a chromoplast. Okay, this is a chromoplast. So let us see this. Now there we have seen several detailed structure of the plastids here. We have labeled also, this is the granum in vertical section. This is a pile of thylakoids. These each sacs are called thylakoids. And this full together is called the pile of thylakoids. Now these are interconnecting bars. These interconnecting bars are called fret. And this base is called, this base of the plastid, chloroplast is called the stroma. So just keep in mind this diagram, only a few things to learn, that is pile of thylakoids and thylakoids. The flattened sac-like structures are called thylakoids. The base is the stroma and these connectors which are connecting one pile of thylakoids to another is called the fret, and these are called granums in vertical section. Okay, is it clear? Just go through the leveling when you are discussing or describing the plastids. If you put this diagram on the right or left side, it will be very easy for the teacher to get a clear picture of this plastids. Now, this is leucoplast. As you know that these leucoplasts are not having any color, so these are white, and this is probably taken from the cells of potato. So these are colorless, and this is colorless, as we have discussed earlier. This is colorless, and this is chromoplast. It is actually taken from red pepper cells. These are all chromoplast cells. This is the cell wall, and this is magnification is 400x. So these are all the three types of plastids which I have taken for your benefit, so that you get a clear idea that how this leucoplast, chromoplast and chloroplast look like, because everywhere you won't be getting these pictures. So just go through it, it is for your benefit only, and make a clear idea of these three types of plastids. And it is very easy. I think if you are having still now any doubt, ask me in my comment box, and don't forget to subscribe to my channel to keep the every time update what I am posting. You can get the updates to do the lesson in a progressive way. Now let us move on to the centrosomes and centrioles. Now in this part, centrosomes and centrioles, this is again, it is present in the animal cell only. So keep in mind the previous was present in plant cell only, and it is present in animal cell only. So centrosome and centrioles are found mainly in animal cell. So what are these? These are small, naked protoplasmic structure present in the nucleus. So and centrosomes consists of two small granules. They contain a small granules which are called centrioles, which lie at right angles to each other. You can see the diagram, which are lying at right angles to each other. During cell division, they migrate to the opposite direction. So in the 10th standard, you will be studying cell division, when you will see that the role of centrioles, how they are helping in the spindle formation, how they are traveling towards the pole during cell division. So they are migrating towards the opposite pole, they are forming a spindle. So that thing you will learn in 10th standard. Now just to learn that they help in cell division. They initiate and regulate the cell division, and they help in the spindle formation during the cell division. So spindle formation is helped by these centrioles during the cell division. So what is the main function of this uh, what is the main function of these centrosomes and centrioles, that we have already discussed, that they help in cell division and they are helping in the spindle formation, as we have discussed in the previous slide. Now, in this slide, we will be discussing about the lysosomes. So, what are these lysosomes? Lysosomes actually, this is a picture of a lysosome. What is it? It is actually a membranous sac which budded off from the Golgi body. Budded off, what is the meaning of budded off? It came off. So it is a part of the Golgi body which came off from the Golgi body. Now these, they actually, what they do, the enzymes released by the rupture of these lysosomes. These are also enzyme filled sacs. So when they are ruptured, they help in the intracellular digestion. What do they help in? They help in the intracellular digestion by destroying and digesting the foreign substances around them. They are destroying and digesting the foreign substances. They are destroying and digesting the foreign substances around them. They are destroying the foreign substances. They are not destroying the known substances. They are destroying the foreign substances around them. And they digest also the stored food during digestion of the cell, because at times when a person is starving, so the digested, they help in the stored food digestion. Okay, they help in intracellular digestion as well as they also help in the digestion of the stored food of the cell. What they are doing that they help, they bring about the cellular breakdown and are associated with aging. What are they doing? They are actually bringing down the cellular breakdown. Cellular breakdown, how is the cellular breakdown being regulated by this? They are bringing it down. So as the cellular breakdown happens with age, they help in the cellular breakdown which are associated with aging. They also dissolve several damaged cells. So several damaged cells are also being dissolved by this suicidal bags of the cells or the lysosomes. So why are they called suicidal bags? So what is the reason they are called suicidal bags? Because they are helping in the intracellular digestion by destroying the foreign substances. So destroying and digesting the foreign substances around them. Hence they are called the suicidal bags of the cell. So the enzymes released by the rupture and of the lysosomes helps in the intracellular digestion by destroying and digesting the foreign substances around them. So hence they are called the suicidal bags. So is it clear to everyone that what are lysosomes, what are their functions, what are centrosomes, what are their functions, and what are plastids? Now we are moving to the last topic or the last type of cell organelles, that is the vacuoles. So what are vacuoles? Now here you can see a central vacuole. So this is actually present, these vacuoles are present both in plant cell and animal cell. So in animal cell, vacuoles are small. So they are present both in plant cell and animal cell. Animal cell, the size is small, whereas plant cell, they fuse together and they form a central vacuole. So this is what down you are seeing is the central vacuole. So this is found in plant cell. This is from plant cell I've taken, but in animal cell it is very small. Now vacuoles are actually, they are what? They are fluid filled sacs present in cytoplasm. In animal cells, vacuoles are small. In mature plant cells, they fuse in the form of large central vacuole. They help to remain turgid. What is turgidity? We have already discussed that to remain hard, to remain turgid. They help us, the plant cell, to remain turgid. And they also play an important role in the elongation of the cells. And in protozoons, especially in protozoons, what are the protozoons? We can say that amoeba and paramecium. In protozoons such as amoeba and paramecium, these vacuoles are, they help in the getting the rid of body weights. So when the amoeba and paramecium, they help in getting rid of their body waste through these vacuoles. Now, they provide a watery environment for the accumulation and storage of water soluble compounds. They help in the providing a watery environment for the accumulation and storage of water soluble compounds. So I think this is all for today. I have completed vacuoles also. In the next episode, I'll be discussing about the nucleus, the protoplasm, and the differences, major differences between the plant cell and the animal cell. We have also to discuss about the cell functions. So this is all for today. This is actually a big lesson. I am dividing it into very small parts and discussing with you so that you get a clear picture of cell organelles, microscopes, etc., etc. So keep track of each and every episode and just give me your feedback, give me your updates, and don't forget to subscribe to my channel. Thank you.